Welcome to the Agrochemical Division of the American Chemical Society's webinar series. Today our speaker is Professor Thomas Mueller, who will discuss off-target movement of dicamba. Tom is a professor in the Department of Plant Sciences at the University of Tennessee. He received his BS from the University of Illinois and MS from the University of Kentucky and his PhD from the University of Georgia. His primary research interests are environmental fate of pesticides, especially herbicides, in soils, water systems, and air. He's published more than well over 100 refereed journal articles in 20 different journals. He served on the U.S. EPA Scientific Advisory Board. He's been heavily involved in the Weed Science Society of America. He's been an editor of a couple of their journals, and he's also a fellow of WSSA. I might also mention he's also the liaison between the Agrochemical Division of ACS and the Weed Science Society of America. Before I turn it over to Tom, I'd like to mention that if you have questions for Tom, you can write your questions in the chat box that should be on your screen, and if there's time at the end, I'll feed those to Tom. If we can't cover all of them, Tom will have information, I think, on who sent the questions in, and he might be able to answer some of your questions after the seminar is over by emailing you. And also, I'd like to mention that the webinar will be on the Agrochemical Division's website at some point in the future, as are all of our previous webinars. And with that, I'll turn it over to you, Tom.
Thank you, Dr. Duke. I want to take this opportunity to say I thank you all for coming. I know this is a busy time of year for many of us. When I approached this webinar presentation, I tried to hit a happy medium between introductory material and some of the latest data that we've got to share, and so hopefully today that's the objective that we're going to try to get to. Today I'm going to talk about the brief introduction of the topic. I have data from 2 field studies, and then just some of the data from our lab studies here at Tennessee, and just a few general closing thoughts about this topic, just to try to bring some perspective on where we are with respect to dicamba. The data presented today is a collaborative effort. I have the joy of working with one of the finest field cooperators, Larry Steckel, the rising WSSA president, who is a fantastic co-worker, and I'm blessed with 2 tremendous technicians, Joe Beeler and Dave Kinzer, who really are the persons who really do a tremendous amount of the work, and I just want to say publicly thanks them for all the wonderful efforts that they have. The views and ideas expressed in this presentation do not represent the views of the University of Tennessee. I work in the Institute of Agriculture. My department, the Tennessee Department of Ag, the Tennessee Soybean Promotion Board, which is my primary funding agency. And today, the mention of a product does not constitute endorsement to the exclusion of others. And so for clarity, several times I've used trade names just to keep this presentation a little bit more clear, I hope. And so we want to make sure that's where we are. This data is unpublished, preliminary, and subject to interpretation. It's in review with several journals right now, and so that's we're just going to put the data out there. My research in this area really goes back to 2009 when I did a project with Monsanto looking at a certain test system to look at dicamba behavior under field conditions. And so really this effort that started all those years ago is the basis for what I'm continuing to do in the field over the many years. From a historical view, I think it's safe to say that herbicides are important in modern reduced tillage agriculture, and the continuous use of glyphosate has resulted in glyphosate-resistant weeds. Now, the first glyphosate-resistant weeds were mainly dicots, and in our Mid-South area, horseweeds and pigweeds were the main source of the lack of control. The University of Nebraska biochemist Don Weeks identified a gene for dicamba-tolerant crops, which really changed the entire agricultural landscape. That trait was later inserted by Monsanto into soybeans and cotton, And seed sales were in 2016. Seed and herbicides sales started in 2017. So that's kind of the background on it. Dicamba is a very old herbicide, first sold in the early 60s, and has many years of actually quite successful use in corn, wheat, turf, pastures, and other situations. However, the new use patterns, in especially soybeans and somewhat in cotton, have really increased the number of acres that are being treated, and many of these farmers had little previous dicamba experience. And this has entered into a situation with some interesting things. Farmers have been used to the simplicity of Roundup Ready crops, which have been a tremendous technology used for many years, and it's the resistance to the weeds that's caused them to change. And on several factors, dicamba is no Roundup, both from an environmental standpoint and from a use standpoint. This picture, which was kindly sent to me by Dr. Duke, shows the estimated agricultural use for dicamba in 2015, so several years ago, but still it's going to be pretty accurate. And the darker the color, the more dicamba there is used. And so basically what we have is in the Great Plains area, this is going to be mainly on wheat, so forth. In the Corn Belt, this is mainly going to be on post-emergence in corn. And if you notice this area in the Mid-South region, this is mainly going to be as a pre-plant application prior to soybean planting to kill a horseweed or mare's tail, depending on what you want to call that weed, because in 2015 we don't have any post-emergent use in this area. But this is a geography where dicamba is going to be central. Now, looking at what it's used on, again, I want to thank Dr. Duke for sending this slide to me. The different colors indicate the different commodities. So corn and soybeans, in the mid-90s, corn, probably Banville, was the primary use of it, and it's gone down some, but still it's a major use, estimated to be several million pounds each year. The soybean use, that's again going to be in a burn down situation prior to plant establishment. So dicamba has been used a long time. There's nothing new about that. One difference with dicamba that was somewhat unexpected is there's often a need for a second application or a sequential application to spray for complete control. And if you're going to have two applications, this would increase the environmental loading. Also, the second application is often made at the higher temperature to larger crops. And so there's more potential to injure sensitive non-target crops, both from the potential of loss into the environment, which I'll show you some data on later, and also there's more non-target sensitive plants available to be potentially affected. I believe sometimes myself as an academic, we kind of forget about the real economic realities of farming today. And there's a large cost differential between the new formulations, which have, my data would indicate, some reduced volatility, and the generics. And there's a really strong temptation with the low commodity prices, and the profit margins for farmers are very tight. There's a temptation to use generics. And we believe some of this happened in 2017, and this year we probably will see much less of that. But this was a factor that may have been a contributing one. Any dicamba discussion on volatility has to have a discussion of the seminal paper by Behrens and Lucian, which was a fantastic work published in Wheat Science in 1979. And just to give a brief overview of it in just the rest of the slide, the main factors they found were the different formulations had an effect on subsequent dicamba volatility. Temperature and humidity, or the environmental factors that is exposed to the dicamba, was a major factor, and plant surface type all affected it. So I could have a different formulation that could increase or decrease volatility. At higher temperatures and lower humidity, you tended to have more volatility. And the plant surface type, when you had surface materials, you could change how much volatility or how much emissions there was. Now I want to just walk through the trade names of what's currently available. The standard generics use is diglycolamine, or DGA salt, which is sold under the trade name Clarity, and many other trade names. So most of the generic dicambas are a DGA salt type of formulation. Monsanto developed a product called Extendamax, which has the DGA formulation. So it's the same salt formulation, and they added what they called trade name Vapor Grip, which is an acetate pH modifier. It's probably potassium acetate at 2% water to weight to volume is what it probably is, but it doesn't exactly state that on the label. And there's a recent article by William Abraham that was been released recently that kind of talks about this specific topic. Fexapan from Corteva, or Corteva, which is a Dow DuPont mergers company, is the same exact concept, but I'll just talk about XtendiMax because that's a product that we use in our testing. A different concept to reduce volatility by BASF was to change the salt, and so the Ingenia is a completely different salt, which is codenamed BATMA, and this different salt has lower emissions. It's also a more concentrated formulation and much higher loading. So as we go through the presentation, I'm going to call them Clarity, XtendiMax, and Ingenia just for the sake of argument, for the sake of illustration. So what happened in 2017? We're still in the introduction report. Well, there was a concern about damage to non-target soybeans, and this was based on what happened in 2016. Just as an example, Arkansas as a state did not allow XtendiMax to be used in 2017. It only allowed the Ingenia formulation. And I'm going to say there was much interest in off-target movements in the mid-southern United States. Now, later in other states, there was occurrences of many off-target observations also. Dicamba has a distinct plant response with upward cupping of the leaves. So it has this very characteristic upward cupping of the sensitive vegetation, and it also can cause epinacity at higher rates. It knocks out the terminals, and that's where you really get your yield effects. Dr. Kevin Bradley at the University of Missouri took the lead, and I've got two slides of his where he showed from 2017 the number of incidents. So this is showing the number of official reports in each state, and there is some variation in how each state counts the numbers. So that's why it's not exactly apples to apples. But if you draw this circle around Memphis, Tennessee, you can see each of these states has a substantial number of events that were officially reported to their state Department of Ag or their Department of Environment, whomever. But if you look at the upper Midwest, there is still substantial numbers of official complaints. And for every official complaint, there's probably a number of unreported events where the neighbors just got together and they settled it without actually filing an official complaint. Another thing that Dr. Bradley did was he said, well, estimate the number of acres that were affected in your state. So if you see some of these acres are pretty substantial, again, in the Mid-South area. But in the upper Midwest, there was also a substantial number of acres of soybeans that were affected, totaling about 3.6 to 4 million acres of affected soybean acres. I've got one slide that's showing that this is from Illinois, and this shows the Illinois pesticide misuse complaints. And so if you look at the long term from the late 80s all the way through, there's about 100 or so misuse complaints in the entire state of Illinois, which has 27 million acres of crops. So it's a major agricultural state. And in 2017, the blue bar is just the dicamba complaints was 2.5 times the amount. So there was, in Illinois, there was some events that was happening to show that dicamba was more of a problem than it previously had been. So now I want to show you two data from two field studies. The first is the effect of surface condition on soils versus plants. And the second study involves the effect of dicamba formulation. All my studies are based on field samples where we collect different sample media, which involves, we call them filter papers, but it's really microfiber filter papers. It's really more than a filter paper. And then we have polyurethane foam puffs, and we store those in a freezer. We later extract them with methanol, and then we assay by LCMS. So the first study looked at what does the surface condition affect on dicamba volatility. So I set up plots with tilled ground, dead plant vegetation in a no-till situation, and live or green plant vegetation in a no-till situation. And we sprayed Clarity, the DGA salt, at 2 times the standard label rate because I didn't know how much emissions I was going to get. And I just want to make a side note, there was a heavy rainfall occurred approximately 12 hours after the application. This is not the exact study that we're spraying, but this is the tractor we spray our studies with. It was applied with TTI nozzles at 20 gallons per acre, trying to follow label instructions as best we can. This is the plot with the tilled soil, and I want to say that this is very high moisture situation. It had rained 2 days before that, and you can see it's a very moist soil situation. This also shows our sampler setup. I've got a close-up of that in a minute. So this is our sampler, which has the filter paper on the front end, which collects particles and so forth. In this part, there is a puff, polyurethane foam plug that would catch any vapors that come through. Excuse me. And this is the venting, because we run these at a fairly high flow rate, and we don't want to have this stream causing any kind of air turbulence. I decided to orient the samplers toward the soil surface in case there was any issues about not having a good collection procedure. The green plant vegetation had a very high amount of plant material. This happens to be mainly common lamb squatters and some other broadleaf weeds. And so this is when I say green plants, this is the green plant vegetation. And we wanted the samplers to be about the same height above the canopy of the plant. So these are sitting on boxes, so they're a little higher up. The data I normally show is going to be this. I converted it to dicamba nanograms per hour, and so this is just a time unit. We collect samples from the time of application for the first 6 hours. So this is basically the morning of application. We collect a unique set of samples and reload all new sampling media at that time. And then we have the afternoon of the first day, 6 to 12 hours, and then we have a new set of sampler media put in, and then we run the samplers overnight, change the sampling media, and then we run the samplers all the next day. So that's how we get our 4 discrete sampling intervals. The 3 general plots indicate the tilled ground, dicamba emissions from the tilled ground, dicamba emissions from the dead plants, and dicamba emissions from the green plants. Now, from Lucian's paper, excuse me, there was an indication that any kind of small amount of rainfall will basically terminate dicamba volatility. So there was a rainfall between these 2 sampling events. So looking at the dead plants, it essentially stopped all dicamba volatility, which was true. But in the green plant material, there was some emissions of dicamba. So when we look at the aggregate, the total averages of the 3, it would appear, based on this data, that whatever the surface condition is can have a major impact on what's going to volatilize later. So if I'm spraying large weeds or large crops late in the season, that could be a factor in why we're seeing more emissions. We measured in a very simplistic way the temperature of the dead plants, green plants, and tilled soil, and the gray bar is the tilled soil, so it was a little lower, which was a possible reason for the lower ones, but no major influence. And same thing with the relative humidity. We didn't see a lot of huge differences between the different treatments, but one of the things we're changing for this year is we're trying to get better environmental data with respect to actually what's happening under field conditions. So the next study was comparing the new to the old formulation. So we set up a trial with the 3 primary trade names: Clarity, Ingenia, and Extendamax. So Clarity is DGA, Ingenia is the Batma salt, and Extendamax is the DGA salt plus the vapor grip. Plot size was relatively large at 60 by 180, or 6 meters by 54 meters. And we decided to put Roundup Power Max in all the treatments because that's the way farmers are going to be using these products. And I understand that that's going to, based on our later data, that could be an important factor. All dicamba was applied at the standard label rate of 0.5 or 0.56 kilograms per hectare. It's the same rate. So this image shows a field shot of our plot sampler. So it was a very high residue situation. The soybeans are about 8 inches tall. Oh, soybeans are in rows here. And just going to the data, it was the same general format. There was no rainfall in this study. We see that in the first sampling interval, second sampling interval overnight the next day. We didn't see quite as much differences. With Clarity had the most emissions, and then Ingenia, and then XtendiMax. And over all our studies, we see this general trend where we have a certain amount of emissions in our first sampling interval. We have more emissions that afternoon. It goes down overnight, and then it goes up the next day. That's more or less a general pattern that we see. So if we look at this in aggregate over all the treatments, we saw that the DGA formulation had the most emissions, followed by Ingenia, followed by XtendiMax. Now I want to remind you that these were all including Roundup Power Max in the mixture, which could have affected it. So the general observations from our field data is that most samples had detected concentrations of dicamba. I would like to say that some of the treatments did not have detected concentrations in some of the surface condition areas with dead plants. We also see that most dicamba lost to the atmosphere per hour was in the first afternoon after spraying, which was the first afternoon. With respect to the surface condition, if there was soil, there was less emissions. And I also want to say that I didn't show the analysis here, but the total losses as a percent of applied were very small. I mean, really a very small amount. And so one of the questions I get all the time is, well, what does this mean for off-target movement? And all along I've said, my objective is to look at the relative effect of surface condition or the relative emissions of the three different formulations. And it's a completely another additional set of studies to say, okay, this is what we're seeing. What does this mean in a biological system? And so that's a completely different set of studies that we're looking for, and that many scientists are conducting those research systems. Well, given the challenges and the variability on the field conditions, many people are using humidomes, and that's a lab study to investigate dicamba emissions. And so I just want to share a couple of data. We've actually run this study, 17 different studies with the different treatments. And so the basic of my studies, I use the air samplers I already have. I can vary the chemical or the surface that I spray, and these are conducted under greenhouse conditions, under variable temperature conditions. So what I do is I just let the temperature, whatever it is in the greenhouse, and then to get the different temperature, I just run the studies multiple times to see what we've got. So this is a visual of the actual setup. So this is the same samplers that we're using, vented to the outside. This is the sampling media, the same tube. It's connected via a pipe which is actually sealed to the top of the plastic. This is the treated area this happens to be a dry soil study. And when we take these trays, they are taken outside the greenhouse, sprayed in an external area. And when that's done, we put a filter paper right here to catch, and we pre-measure. So every tray gets measured what was applied to it, and that gives us an untreated area right here to put the, this is a temperature and relative humidity sensor. And so that's how we actually run the studies. So I'm just going to show just some general results of the first one. In the first studies, and I'm not showing the individual data, but just an aggregate of the first 10 studies, temperature appears to be a major driver, with higher temperatures coincident with more emissions. We have come to we measure the pH of all our treatments, and when you have a base dicamba treatment, the pH is about 6, and when you add the Roundup Power Max to it, it drops at about 1 pH unit. But within all the different measurements, the pH similars are similar. And our results in the field and our humidomes look very similar in the patterns. So I'm just going to show you one data set. So this happens to be 3 different formulations applied to dry soil. The average temperature over the entire study was 85 degrees or 29.5 degrees centigrade. And so this is the pH of the spray solutions after adding the Roundup Power Max, so it's down to about 5. Looking at the Ingenia or the XtendiMax or the DGA formulations, in the humidomes we run 0 to 12 is our first time interval, and we have 12-hour intervals for up to 60 hours. So in the middle of the day, we have the greatest concentration. Overnight, it goes down, it goes up the next day, and so forth. And this temperature plot, which is on a 30-minute time step, this temperature spike is coincident with this peak, and this smaller peak is coincident with this temperature valley. And so we're going to do the correlations to show that temperature is a major correlating factor with the dicamba emissions that we're seeing in this test system. Another type of study that we do is we spray a common herbicide, in this study, Ingenia plus Roundup Power Max, and we change what's in the tray. So we have dry soil indicated by this set. We have wet soil with no irrigation in this series of data points. We have dry soil with irrigation indicated here, and then we have wet soil with irrigation here. Now, the irrigation was applied, basically a ½ inch of simulated rainfall was added to the top of the tray after the 12-hour application. We collected the samples and irrigated those respective studies. So when the soil remained dry the entire time, we had the highest temperatures, the lowest humidities, and we had the greatest amount of dicamba emissions. Where we have the wet soil, we had intermediate amount of dicamba emissions over the whole time course. Where we had the dry soil or the wet soil with the added irrigation, we had similar amounts of emissions post-irrigation. And so to show the effect of irrigation, this is the dicamba recovered on all nanograms per treatment plotted against hours after treatment. So in the dry soil treatment, the emissions continued to increase, and the slope of this line is pretty substantial compared to once you have irrigation, the slope of the line is much less, indicating, as anticipated, that the dry soil, when you irrigate it, it greatly reduces dicamba emissions in subsequent sampling intervals. The same data exposition looking at wet soil shows that there really isn't that big an impact on the dicamba emissions between wet soil and if you irrigate it. There's a little bit of difference, but not much. So this implies that it's not really some sort of a pre-emergent incorporation factor that's moving the dicamba into association. It's really more of an indirect temperature relative humidity effect. At least that's one way we're looking at it. Now, this is a really busy figure, and I'm going to try to go really slow and explain it, and I apologize for the complexity, but what we have here is we have the temperature of this respective treatment against the time course of our study. So the dry soil is the green circles, the red is the wet soil, black is dry soil that's irrigated at 12 hours, and yellow is wet soil that's irrigated at 12 hours. So if you look at the dry soil and dry soil plus irrigation, the green and black, up until 12 hours, these are the same treatment because it has not been irrigated yet. And once you get to 12 hours, at that point, then they separate. And the green one, the green data, which has no irrigation, has a higher temperature, especially in the middle of the day and overnight. All the others, which have been either wet soil or irrigation, their temperatures are basically all very similar over the time course. So that's the temperature data. So that when it's dry, you have higher temperatures at several points. Now looking at the same data setup for the relative humidity, and here again, this is the relative humidity against the time course. The green data is the dry soil over the entire time course. Red is the wet soil over the whole time course, and the black and yellow are the dry, wet soil, irrigated, and so forth. So let's take a look. The dry soil, here again, these two lines are identical until they're irrigated. And once it's irrigated, then it becomes a much higher level of humidity, which makes sense. The humidity level for the entire time course is much lower in the dry soil, which is completely normal. The wet soil, based over this data, is slowly drying out in the second half of the study because we don't add any additional moisture. It appears that there's going to be a lower humidity, which makes sense because the soil is drying out, especially these high temperatures. Now, when I first looked at this data, these spikes, I don't really like seeing these, but I can tell you what's going on is whenever they take a sample, they turn off all the samplers, and then this is a spike in the humidity, just happens to catch it while the samplers are being changed out. and we have 10 samplers that we're changing out. So that's what this is, a temporary spike in the things. And we've changed our procedures, so we are trying to have more rapid turnout of the different samplers. But the data is what the data is. So this is showing the differences in humidity, and there's a substantial difference in humidity, which may be a factor in why we're seeing more dicamba emissions in the dry soil. Its humidity could be a factor. And this is consistent with Aleutians and Barron's data. Okay, so that's the data. I want to just go to a few changes that have happened in 2018 to get the update. Many states have made it a restricted use product. You must use the new formulations. Many of the label changes involve time of day restrictions where you want to avoid inversions, and I'll talk about inversions in just a minute. You can use only low drift nozzles. You have to keep the spray boom less than 24 inches from the target. Wind speed, it has to be more than 3 miles an hour, but less than 10 miles an hour. And I think in some jurisdictions it's 15, but I'm just saying there's an upper and a lower limit of the temperatures. There's buffers based on the field edge. I mean, and by the way, the buffers are based on the ESA, or the Endangered Species Act. And there's mandatory training. Some states added more restrictions, and I'm not going to get into the details of that, but I know several states added cutoff dates, they added temperature things, and so forth. So there's also more restrictions. There's much of the concern about spraying dicamba in 2018 was not spraying into or during an atmospheric inversion. An inversion is very calm air with essentially no wind, and it's technically defined by the reverse temperature gradient. But the primary outcome is less dilution of the particles, whether they're aerosols or vapors, and there's huge debates about what is actually moving in the atmosphere, whether it's an aerosol or it's a solid particle or it's a vapor or so forth. But the field observations would indicate that spraying into an inversion could result in more off-target injury, and much of the label changes are based on not applying into or during an inversion. So an inversion, in a normal situation, you have hot air near the surface, and the higher you go, the cooler it is. And there's quite a bit of turbulence in this situation. In an inversion, you have very stable air at the soil surface, and there is not much mixing. And so when you have unstable air, as you would in a 3 to 10 mile an hour wind, you have diffusion with good dilution. So if you do have any emissions in the air, it's diluted downwind. If you're in a stable situation, dispersion dominates with poor dilution, and what you can have is whatever is emitting can move a half a mile or a mile pretty easily and can affect off-target sensitive vegetation. So we've got these new labels, and many of the new auxin herbicides have new labels. And from an agronomic perspective, to follow the label completely and still kill the weeds is almost impossible. We have rapidly growing weeds. Palmer pigweed or water hemp are rapidly growing weeds. There's a very short time interval to spray. And from a legal perspective, and I'm not a lawyer, but once an application is off-label, then the liability shifts away from the registrant and to the applicator farmer. And this has set up an issue. And so I just, a colleague from Tennessee sent me this quote, and it says, "No law is of value without voluntary compliance." And what I mean by this is that we don't have enough inspectors to police everybody spraying dicamba. And so whatever is used, however we use dicamba in the United States, we've got to have voluntary compliance, good stewardship of this material to avoid any potential off-target movement. And we need to have a better year in 18 than 17 to have success with respect to the use of this product. Because the truth is, dicamba has become a divisive issue, and there's two general camps. There's Camp A that says use it, it's great weed control, and Camp B, do not use it because of off-target movement. Well, it's really a little bit more complicated than that, because on Camp A, you've got cotton and soybean farmers who love the technology, who really need the technology, and some companies. And then you've got the second camp, which are Liberty Link growers, conventional soybean growers, vineyards, homeowners, organic producers, tobacco producers, and everybody else. And so it's really it has become somewhat of a divisive issue in some quarters. Now, just a few closing comments. I've got 3 more slides left. there's a new primary registrant for the traits. So if you didn't know, Bayer has bought Monsanto, and they're in the process of making a major change on that. Dicamba symptoms are unique to plants, so you really can't say it's a virus or something else because of the specific symptomology that it causes. One of the things I don't think a lot of people are saying is the toxicology profile of dicamba is excellent, and there's virtually no mammalian major toxicity of dicamba. I'm not saying you can drink this stuff. I'm not saying that, but I'm just saying from a mammalian toxicity standpoint, dicamba has a very clean profile. Another thing nobody's saying about this is there are people who are using this Extend technology and have an excellent broadleaf weed control, and they're the cleanest fields we've had in 5 years. So the benefits of using this, where I've got Palmer pigweed and my soybeans are cotton, is absolutely huge, and that's why the people have such great interest in it. For those that don't work with dicamba every day, you may not know this, but when they did the label 2 years ago, the current EPA label only runs until November 18th, and at that point it sunsets. So in the absence of any activity by EPA, the dicamba the use will cease because the label runs out. Much of the talk about the training and everything is to talk about spraying into atmospheric conversions, and I think it's interesting that we're seeing right now with really not volatile products in rice in Arkansas that they're seeing some problems spraying into inversions, and has nothing to do with dicamba whatsoever. I just want to say I thank you all for coming today. The technicians, my technicians, Joe Beeler and David Kinzer, are fantastic. My students, Trey Clark, Luke Schaffner, Abby Vickers, who do such a fantastic job in everything that we do. I want to appreciate the funding by the Tennessee Soybean Promotion Board and the future funding from the United Soybean Board. I just really sincerely appreciate visiting with you today, and I tried to get through this fast enough to leave time for questions. So, Dr. Duke, that's what I've got.
Thanks, Tom. There is plenty of time for questions, and again, there's a box there. Should be a box on your screen for sending questions in, and I see we've gotten quite a few already. Let me start by asking a few of these. Mark Zinn asked the question, what is the wind speed, or what was the wind speed during your study, and do you think it had any effect?
The samplers, Steve, do you think I should repeat the questions or not?
It's up to you, Tom.
I'm asking you.
Okay, yeah, go ahead.
The question is, what was the wind speed during my field studies? Well, there's two factors I want to say. First off, the sampler is located in the very middle of the trial. So I'm not measuring off target movement per se. I'm just trying to get the relative emissions from the field surface. Most of the time we had, 3 to 5 miles an hour, but there was some gusts in other studies where it get up to a little higher. But I would say none of these were really exceptionally windy trials, but that's the data that we're going to put out there.
Mark has another good question. Why is there a lower limit on wind speed for the label?
The question is, why is there a lower limit? Because you don't want to be spraying into an inversion, because if you spray into a really still air mass, then you're not going to have any dilution of what you do spray if there's any aerosols that are coming off your field. So they want a little bit of wind to, what you're really doing is not spraying into an inversion.
Okay. Here's one from Bob Scott. You may not want to try to answer. It's essentially, what's the concentration or parts per billion of dicamba you need to cause symptoms in susceptible soybeans?
The question is, how low can you actually see symptomology with soybeans? And there's been several studies that people have done, and it's an exceptionally low number. And to give you the number, what I've heard is 18 nanograms per cubic meter. I mean, if you want a number, I'll give you a number, and it's 18 nanograms per cubic meter. Now, I don't remember where I heard that number, but it's going to be a really small number. And I think it's safe to say that soybeans are an exceptionally sensitive, hypersensitive species. And Bob Scott knows that's a really touchy, a very cogent question, and so it's a good question.
Okay, and I've got one, Tom. How much damage can you get to soybeans before you affect yield?
The question is, how much damage can you get? If you have damage early on to vegetative soybeans, the soybeans are a tremendous plant to recovering and really having no yield loss. If you knock out the terminals and you have substantial you knock out the terminals, that's when you really get injury. I didn't get into it today, but another thing that Dr. Steckel and many weed scientists are saying is many times you get the soybeans drifted on once, okay, it'll regrow it. But what happens is you'll have sequential drifts. Remember I mentioned early on where you have sequential applications. So you get drifted on June 15th, okay, I'm not too worried. I get drifted on again July 1st. Now that sequential application, that's what's really doing the yield, any potential yield losses. So that being said, our estimates in Tennessee is that of all the acres affected, only 20% had a yield loss, and the average yield loss from that year was approximately 10%, 15% of the yield loss. But if you go back to remember last year, we had absolutely perfect weather conditions. And if we didn't have the perfect weather conditions in Tennessee, I believe we would have had substantially more crop injury than we actually noticed. Does that speak to your question, Dr. Duke?
Yeah, I think so. Laura McConnell sends this one in. How would you rate the importance of drift versus volatilization as a cause of crop damage effects?
Okay, the question is really separating particle drift from vapor drift. And it's going to be very individualistic on the application. And I think the challenging thing that we see is there are times when large fields are affected and there's no apparent particle drift pattern. A particle drift pattern is more a gradient from the source of the field, normally adjacent to the treated field, going away from it. And so she has asked a very good question to say, well, we really can't say, is it aerosols or is it vapors and so forth? And I mean, my samplers detect dicamba, and I can't tell the difference between a vapor and a particle of soil or a particle of water, or, you know, so I think it's a really good question. So, and the answer to her question is it's going to depend on the actual application situation. And the frustrating thing for many applicators is there are people who've done everything correctly by the label, and there is still apparently off-target movement, and that's the frustration for many people.
Okay, this is from Logan Greyer. In your field studies, he says he knows that the goal was symptomology data, but did you see symptomology in the surrounding sensitive soybeans?
We really didn't look for it. The way we originally do the studies is we're not really focusing on it. And I would say in answer to his question, since it's a good question, is that we didn't really see a tremendous amount of damage outside the treated area. But the truth of the matter is we're not looking 14, 28 days later when it would be most likely to be seen. And there's a lot of people, I mean, and I could name 5 scientists who are doing work in that area about they are doing that exact study, that they treat an area and look at the outside treated area. And so there's a lot of good data out there. And that's not the focus of this study, because we have enough jobs to just keep running our samplers and getting our relative amounts of dicamba emissions.
Okay, here's another one from Bob Scott. Do you believe that you're seeing reduced volatility overall? Let's see, do you believe that you're seeing reduced volatility over all the formulations, or just delaying volatility over time?
Okay, the question is, am I seeing delayed volatility or reduced volatility? We see reduced volatility. I mean, I firmly believe that the ingenious salt has lower volatility or lower emissions under field and lab conditions than a DGA salt. And I firmly believe that if you add potassium acetate or whatever acetate modifier to DGA, you do have reduced emissions under field and lab conditions. Now the question is it low enough that it gets it below a biological threshold? And that's really the next question we're going to look to answer. So I think it's reduced on the first sampling intervals and the later ones also.
Okay, here's one from Gabriel.
Hey, Steve, no more questions from Bob Scott. He's done enough. So no more Bob Scott questions.
Okay, this one's from Gabriel Rothman. And Bob just said, okay. Gabriel says, did you look into tighter air sampling intervals, 1 or 2 hours, for instance, looking for rapid changes with temperature and relative humidity.
Yeah, that's an
Because it's
excellent.
saying moisture and temperature conditions rapidly change after rain events and after temperature inversions.
Yeah,
So
I once you start doing a new test system like this, at least due to us. I mean, there's other people who've done this kind of projects. There's all kind of different ideas you can have, and we're trying to hit a happy medium between enough fineness in the data to make some significant observational data. We always struggle with, if you take a real short time interval, you lose sensitivity and you get a lot of non-detects. And in the paper that's published, the 2013 paper, we actually had different time courses in there and had a little bit finer sampling interval. But yeah, no, we did not do those timing intervals, but that's something we can look into. Also, to do field studies, I mean, we didn't plan to have that rainfall. I mean, it is what it is. So we had the observations based on that. But I agree that his point about where you pick your intervals is a really interesting and challenging aspect of this type of research.
Okay, Steve Cryer asked this question: would altering the application timing to when temperature is cooler be the major benefit and why didn't the experiments go beyond 60 hours since you were still getting quite a bit in your last sampling interval?
Well, okay. Well, is he talking field or lab data? I don't know.
I'm not sure.
Could you read that first part of the question, just the first part?
Let me read the whole thing. It says, looks like volatility may be at play. Would altering the application timing to when temperature is cooler be the major benefit? I'm not sure what that.
Yeah, well, we spray it first thing in the morning, whether it's a humidome or a field study where the temperatures are cooler. And so, okay. In fact, the companies don't like it because I'm spraying at 6 o'clock or 7 o'clock, and I spray into an inversion, so I'm off label, and they yell at me for that, which is fine. They can yell at me, that's fine. But so we actually are spraying when the temperature is cooler, and one of the reasons under field conditions why we have a 0 to 6 sample is because we want to say, in 6 hours, surely all the aerosol droplets are going to drop in 6 hours. And if I have any wind at all, I mean, if I have a 1 mile an hour wind, my aerosol droplets are not on my samplers anymore. They're gone. They're downwind. Now, with respect, the other thing is we're seeing so much more volatility. Yeah, I mean, it doesn't matter what it is at the time of application. It's the temperatures and the humidities that afternoon or the next day that matters. And we did run humidomes up to like 7 days, and it still didn't have a diminution in volatility after 7 days, which I thought was staggering, and I didn't show that data. And I don't understand that at all. The other thing is these student workers got a life, and I can only work them so much. And, so you run studies, there's just logistical reality about running them. So we've settled on 60 hours as kind of a happy medium to run the studies.
Okay. Lou Mayer asked this question, for dicamba residue analysis, do you have an established LOQ for the puff sampler?
That's a really good question. The puff substrate is difficult to work with, and so when we did fortification studies, we were getting good recoveries, but I'm going to revisit that some more because when we do the humidomes, we see nothing on our puffs. Everything's on the microfiber filter paper, and we're doing research to try to have like a pre-filter in front of it to try to figure out how we're going to do that. But when I did my recoveries, I had good recoveries, but that was just a straightforward fortification extraction system. And I think he asked a very good question. Are you actually capturing vapors, and what is the recovery of the vapors that we're doing? So we are currently doing studies to kind of look at that some more. I think that's a very key question. And the puff matrix, I mean, just polyurethane foam as an analysis, it's just a mess. I mean, it's very challenging to run. So I think he asked an excellent question, and in probably reports over this winter, I'll probably have better update on that specific answer.
Okay, Randy Weintraub has this question. Do you have any data on the effect of soil type on volatilization?
We have not, because we're doing everything with a Sequatchie silt loam. I know there's a group of people that really feel that the soil is a major factor. And I guess from a field standpoint, I'm looking at it as when I'm spraying these field plots and there's so much plant material, under field conditions, I'm having a hard time seeing that what soil is underneath all that plant residue really matters. Now, that being said, there's clearly times, I think, where there's a lot of soil present, it could be a factor, and that's something in our system. We could easily change the different soil types in our humidome. So, I mean, we could do that very easily. So that is a researchable topic, but I don't have any data on that right now.
Okay, Shanique Grant is asking, do you have any plans to measure dicamba off the field in future studies?
Yes.
Okay. Okay. And Douglas Stevens comments, no mammalian or human metabolism is known for dicamba, but human health impacts can also result from non-human metabolism that occurs in the gut. Have any studies been performed on adverse human health consequences of dicamba exposure as a result of gut microbe metabolism?
I'm going to defer that to you, Dr. Duke.
I'm not a toxicologist. Keith Solomon might have been able to answer that, but
yeah, I would say that
outside my
given the dicamba use labels in especially turf where you're going to have child exposure, I would venture to guess. Now, with respect to his exact question, I don't know. I'm not gonna comment because I do not know that specifics. But I think it's safe to say that if dicamba had appreciable mammalian toxicity, I think we would be having a very different discussion on the movement off-site than just plant damage. I think that's a safe statement to say.
Okay. And Cheryl Cleveland, I don't think I've asked this one yet. Based on your research, is there any benefit to spraying in the evening?
Based on the spraying in the evening is off-label because you can't spray after a certain time because you don't want to be spraying into an inversion. And so we did do some of that in the paper, and it showed that you could potentially, from a research standpoint, I want to say this really clearly, the research standpoint, the idea was you spray it at night, it absorbs into the plants all night, and you have less ability to volatilize the next day. That's kind of what the paper said. In fact, I think that the paper says effective time of day it's in the paper actually. So although technically that may be a possible way to do it, they don't want to do that because the risk of spraying into an inversion and getting aerosols, particle drift overnight is of greater concern to the registrants than any vapor drift. I think that's the way I want to answer that question. But okay. I will say that time of day is a major factor. We saw that in previous studies.
Okay, Laura McConnell has another question. Can you calculate the fraction emitted of what was applied to the field?
Yeah, it's a very small percentage. I mean, it's depending on the assumptions you make, and it's just, it's going to be a very low percentage, and I had that number in there, less than 0.01%. And so it just depends on the assumptions you're going to make. But I think if based on my analysis, if you think 2 or 3% are coming off, that's not true at all. I mean, it's a very small percentage what I'm measuring.
Okay, Keith Wing is asking, do you think it might augment your work to place potted soybeans at different distances from the treatment plot and monitor at different times?
Yes, in fact, we're doing studies right now where we're adding that. I made the philosophical decision that I was not going to do that. I know several other scientists are doing that, and in fact, they were doing, basically that's what they were doing. And there's a whole body of research where that's exactly what they're doing. I mean, Jason Norsworthy, Brian Young, several others are doing that exact study where they're using the bioassay species of soybeans and they're putting plats in and out and so forth. And we're doing some standard protocols where this year we'll do some samplers where we'll have samplers and then we'll also have the bioassays. It's real easy to contaminate the soybeans. It's a lot of logistics of trying to get all the soybean plants in and out of the field without contaminating them and so forth. But there's no doubt that the bioassays, the soybeans, are very sensitive. So I think it's really good for qualitative data. I like my samplers for quantitative numbers to get an actual relative amount. But yeah, I think there's a lot of people doing the soybean bioassay route.
Greg Kruger is asking, what percentage of what's emitted is coming off as volatility versus drift? particle drift?
Well, the samplers I'm running detect dicamba, so I'm not going to say whether it's aerosols or vapors per se, but I will say that based on the sampling interval, I'm having a hard time believing that any aerosol droplet is present more than 6 hours after application in my field system. And so it's not, I don't think it's aerosols that are emitting from the nozzle. I think it's something that's hit the target, and by whatever format, whether it's an aerosol that's re-emitting or re-evaporating or a particle or a vapor, that's what I'm detecting, especially the second, third, and fourth sampling intervals. The other thing is in the humidomes, there's no aerosols because they're sprayed outside the greenhouse, and they're transported in a way that there's no aerosols that are coming with them. So I know Dr. Kruger's an aerosol guy, but it's not aerosols in this test system.
Lee Van Witchen asked this question. Are there any plans to look at the emission from different, you saw higher levels emitted from green leaf surfaces? Any plans to look at different species, corn, soybeans, or certain weeds in terms of whether there's a difference between different leaf surfaces and emissions?
Yeah, I think we're planning to do some of that in the humidome setting. It's difficult and challenging to do field studies because it just takes so much space, and there's so many other factors. But we're planning to, and we have done some of that under humidome conditions. So yes, we're planning to look at that. Okay,
thanks, Tom. We've gone almost an hour, and I think we've covered the topic pretty well. I really appreciate all the many questions. We didn't cover all the questions that came in, but I think we've covered 90% of them. And I think most of you know how to get a hold of Tom if you still have more questions. So please email him if you have more questions. Thank you very much for your participation, and look forward to hearing more about this topic in coming months and years. Thank you.